Dr. Kishor Upreti, MD, pediatrician, senior consultant pediatrician, and he comes from his own expertise center of pediatrics, Lily Kid Care Clinic. And as everybody has been familiar with the topic what we are going to discuss today, it's genomics, behavior and more. The session will go very interesting only if you participate so actively. So make sure that this session will go so lively because through your active involvement. So in between, I should also introduce our very respected Surinaran Paudel sir. In fact, I had invited Surinaran Paudel sir for the session on economics, and there was some technical issue in the correspondence. But we are very honored to have him too in the session today. I hope sir will manage his some time today and be a participant today. So, Dr. Sir, please let's lead the session with your very interesting topic of discussion: genomics, behavior, and more. Please, over to you.
Thank you, Mr. Sir. I'm very privileged to be here. It's kind of a different kind of experience for me. I'd like to welcome Principal Sir, respected Surinaran Sir, esteemed faculties, and all the learners from MBA session. Because I'm from a different kind of field, and I guess this class composes of people from different background, maybe science, technical background, maybe accounting background, maybe economics, or maybe many more. So with the advent of new technologies and innovations, we have integration of different faculties, different field of subject into another. So it has a different kind of implication. Actually, I'm not more into corporate sector or organizational science, but as a medical person, as a student of biology, science, and medicine, maybe I hope I'll be able to give you some insight into the basic part of this faculty, like gene, genome, and how it affects behavior and how it relates to your faculty, like organizational behavior or managerial and corporate sector. So I'm very honored to be here, and I'd like to thank especially Mr. Susan Karti, who is a long time acquaintance of mine and one of my brother, very talented and very scholar person, as I've been knowing him for more than 2 decades. So thank you, Mr. Susan, for this opportunity. So I'll start with my introduction. I'm Dr. Kishor Upreti. I'm specialized in pediatrics. I'm a consultant pediatrician. I've been practicing pediatrics for 11 years now, and I have worked in different government, private sector, and I had an opportunity to work abroad in Maldives as a pediatrician for a year. So this is all about me. So we'll introduction session like it will be better so to know each other before we start really on the topic. So we start with introduction.
Mother Suraj Sreshta, the pilot is
I know, actually I have just kind of I've seen somewhere.
My profession is software engineer plus my software business manager. I will say MBA planning. Nice.
Hello Dr. Very good morning. This is Shativan. I belong to Zanapur, and I am fully occupied with the corporate life as a production manager in a multinational company. And recently I have joined this session, and I hope our batch is going to enjoy your sessions and insights. Thank you.
नमस्ते, मेरो नाम लेखा सन्दिप हो। मेरो घर चाहिँ सुदूर पश्चिम मध्य जिल्लाबाट अहिले यहाँ एमबी बैठा छु।
गुड मर्निङ, म विश्व बराल। मेरो घर चाहिँ भरतपुर अफिसबाट अहिले चाहिँ म सेन्ट्रल इङ्ग्लिस बोर्डिङ स्कुलमा चाहिँ एकाउन्ट एन्ड एडमिनिस्ट्रेटिभ अफिसरको रूपमा चाहिँ कार्यरत छु।
हजुरलाई गुड मर्निङ, मेरो नाम सुजन भैया हो। म चाहिँ अहिले प्रज्ञा एकाडेमी मेघालीमा आबद्ध छु।
मेरो नाम अङ्किता सिलाइ भण्डाराबाट अनि अहिले चाहिँ म रिसेन्टली आइएमपी
नमस्ते सर, मेरो नाम चाहिँ रश्मि खत्री, म चाहिँ युनाइटेड अडिट इन्सुरेन्समा जब गरिरहेको छु।
Good morning. Myself Muna Thapa. I'm recently graduate from Tribhuvan University and today I'm here.
Continue study of it.
नमस्ते, मेरो नाम रम्या सिन्हा, म चाहिँ यहीँ बस्छु अनि अहिले चाहिँ म एउटा मल्टिनेसनल कम्पनी चाइनिज एग्रोबेस्ड कम्पनी एज अ फाइनेन्स डिपार्टमेन्टमा काम गर्छु।
Hello sir, good morning. My name is Lakshmi Kumar र अहिले चाहिँ म विमल कार्की एन्ड एसोसिएटमा चाहिँ एकाउन्टिङ प्र्याक्टिस गर्दैछु।
गुड मर्निङ सर, आइ एम समीक्षा बराल, म चाहिँ आइ फुल टाइम स्टुडेन्ट।
मर्निङ सर, my name is Nikita Kamlari from Northwest East. म चाहिँ फुल टाइम स्टुडेन्ट।
नमस्ते डक्टर, म चाहिँ आरती सालुज।
म नमस्कार बाबुलाई म चाहिँ आरती सापकोटा भदौर 5 बाट करेन्टली एउटा मनोकामना ग्रुप अफ कम्पनीजमा एज अ अकाउन्ट अफिसर काम गरिरहेको छु।
हेलो सर, नमस्कार, मेरो नाम चाहिँ गिदान बस्नेत, म चाहिँ भरतपुर 13 गीतानगरमा बस्छु। म चाहिँ बिसिए स्टुडेन्ट, अक्सफोर्डबाट पास आउट भएको छु।
This is Dilip Kovic. I'm into IT company for last 8 years. We do design, develop, and promote businesses in Chittagong. So if you want anything for your clinic, we can work on that.
That's the theme of session 9. Okay.
नमस्ते, भेरी गुड मर्निङ सबैलाई। म सूर्यनारायण पौडेल, पेसाले प्राध्यापन गर्छु वीरेन्द्र क्याम्पसमा। विगत 12-14 वर्ष इकोनोमिक्स डिपार्टमेन्ट हेडको रूपमा काम गरेँ र चितवन जिल्लाका विभिन्न कलेजहरू प्रेसिडेन्सी, अर्किड, अक्सफोर्डदेखि लिएर धेरै कलेजहरूमा आलुमारी पढाएँ 12-15 वर्ष। अहिले सबैतिर प्राइभेट इन्स्टिट्युसन छोडेर बसिरहेको छु र म इन्टरप्रेनियर पनि हो। रोज गार्डन पनि चलाइरहेको छु। अहिले उद्योग वाणिज्य सङ्घलाई एउटा एसिस्ट पनि गरिरहेको छु, एक्सपोर्ट र इन्भेस्टर पनि हो। स्टक मार्केटसँग नलेजहरू पनि सेयरिङ गर्ने गर्छु।
Thank you. By the way, my name is Man Adhikari. म यो कलेजको BBA HOD प्लस प्रोग्राम कोअर्डिनेटर। म 7 वर्षदेखि चाहिँ कलेजमा आएको छु। थ्याङ्क यू।
नमस्ते सबैजनालाई, मेरो नाम विवेक लामिछाने, फ्याकल्टी मेम्बर भ्याली स्टेट कलेज।
नमस्ते, मेरो नाम निरञ्जन कार्की, म फुल टाइम ट्रेडर, पार्ट टाइम टिचर।
नमस्ते, मेरो नाम विनोद नेउपाने, म फुल टाइम फ्याकल्टी इङ्ग्लिस डिपार्टमेन्ट भ्याली स्टेट कलेज।
Thank you everybody. Mr. Susan Karthi needs no introduction anyways, you all know him. So, before going into the topic, I'd like to request that don't get confused with terminologies. I'll try to explain in very basic form. सबैजनाको साइन्स ब्याकग्राउन्ड छैन, I know, but whatever background you come from, just we'll try to explain it in the way that we can understand very easily, and don't get very occupied with terms and terminologies and all, because I don't think this is your very syllabus session kind of thing. This is kind of thing which is like explorative and having new knowledge and having expanding your knowledge into different sessions. So let's start with the topic, genomics, behavior and more. So I have some. So this will be the overview session. We'll talk a bit about history, how it started and all, then basics of genetics, how this genetics have developed and how genetics works for us, and then the core of the topic, behavioral genetics, and then we'll go on to genetics in practice, like how this genetics actually can be implemented in how day-to-day practice as a administrator. So this person, we all know him, heard him this is not knowing by face. We have heard his name because this is the basically we study. Everybody of us have studied in high school, 9th, 8th, 9th grade. I think at our time we studied that time. This is Gregor Mendel. He is considered as a father of genetics. At that time, 1800 AD, 1813 he was born, and somewhere around 1840s and 50s he was there. He didn't have very sophisticated tools, but what he observed was how characteristic of one generation passes to the other generation. So he noted that, and he did experiments with pea plants. So I think we all have done that calculation in the box, capital T, small t, and then how offspring can be, how many possibilities are there, if there are dominant character, how it affects, if there is recessive character, how it affects. So without any molecular technology, any advanced technology, Mendel has conducted such kind of experiments so that he can know the characteristic that how one characteristic can pass from one generation to another generation. And he has founded the concept of genetics, so he's considered as father of genetics. And then I'd like to write something there. We have a lot of words, sometimes we get confused. So we have some word called gene, and we have, I think it is legible, पढ़ना सकिन्छ है, genetics अलग ठुलो लाग्नुपर्छ कि, we have genome, we have genomics, then we have chromosome. So a lot of terminology we have been hearing. I think we have all heard of this term: gene, genetics, genome, genomics, DNA, chromosomes. So what is the difference? Actually, the gene is a basic structure of the genetic material. So it consists of 3 base pairs. Okay. So I'll again explain in the next basics of genetics. So you consider it as a unit of that genetic material. That is gene. Study of gene is genetics. So genetics study particular gene in focus. So it is not studying whole thing at a time. Okay. And then in contrast, genomic study, genomics. This genomics studies, genome is a group of genes. So there's importance. I'll come to that later, because single gene. If you study single gene, this gene is going to act in a way. It has a different importance. If you study same gene in a group, then it has interaction. One gene affects the other one, then other one in turn affects the another one. So this is a broader term, and the most complex part is genome and genomics. Gene is just simply a set of unit that we are studying that is in particular in focus. Okay. So and DNA, when genes are packed into the packaging, like we have unit. Suppose we have a unit of biscuit, and then we pack it into the bundles of dozen. Then again we pack it into that dozen packets into another dozen. That kind of thing is DNA and chromosome. So if we pack that genes into the groups and bundles, that becomes DNA. What we see represent is like double helical structure like this. This is DNA. And DNA is further packaged into very compact form. And with the help of other proteins, we call histones, and there are other nucleotide, and then hexose sugar. This is a kind of structural protein. Then this is further packed into another compact structure that is called chromosome. So it looks like this. It looks like this. Okay. So gene is just a unit of that particular structure, and we study gene means genetics. Genome is study of lot of genes and their interaction, how that interplay with each other. DNA is packaged form of genes, and chromosome is that is further packaged into another form that is called chromosome, which lies inside nucleus. father of genomics.
I have a small curiosity. I hope most of us have also heard about the RNA.
RNA.
So how RNA related to the DNA?
So I'll come a bit later with that. RNA, it has very important role. So that comes into the play when this is translated into food. When gene is working, then RNA comes. So I'll explain that in a while. Okay. So this is Mr. Sanger. Sanger is one of the few persons who have won multiple Nobel Prizes in same field, biochemistry. He was actually biochemist. So why he is important is that he has developed, Mendel has described that gene exists and then character is passing from one generation to another generation. This guy has developed some really good technology so that we can study molecular, we can study genes at molecular level and we can see the effect of gene, how it is translated. So he was given Nobel Prize for that, and we are considered father of genomics. That was the father of genetics, and he is father of genomics. And then one more person to mention, he is also from around late 1800s. Galton, Mr. Galton, Sir Galton is considered to be father of behavioral genetics. So he was considered, he is the one who proposed the theory that behavioral characteristics are inheritable, like it passes from one generation to another generation. So now, as we have raised question, basics of genetics. Basics of what is gene actually? So we have talked about gene. Gene is just a sequence. Gene is just a sequence, structural sequence, which determine complex physical, physiological, immunological, and a lot of things, behavioral, and then emotional, intellectual entity of the living being. Not only human. We'll more focus on perspective of human in case of behavioral genetics, but it affects everything. It affects everything. And another thing to consider is not only gene affects our functioning, but our external environment also affects the function of gene. So there is two-way interaction. It's not one-way interaction. And there are genes which are easily affected by environmental like stimulus, but there are some genes which which is intact throughout the generation. And there comes another theory of it where evolution lies, because if genetic changes and genetic external stimulus has not changed the genes, then we would have looked like somewhat like Ramapithecus or Neanderthal, but we are different now, because there are a lot of things happening with genes, because genes which are more strong properties keeps on passing to the generation, while with weaker properties keeps on vanishing. So we'll see. We have some of the students from science background also. So we'll see what is gene. Okay. So gene is simply structure. Okay. So we have. So we have something called nucleotides. Okay, nucleotides. Okay, nucleotides are just simple molecules. Okay, so these molecules are adenine. Okay, cytosine, and then uracil is in RNA as you have, and guanine and your iosyl. Okay, so just these are letters. Okay, but these are nucleotides. So if there is sequence like A, C, G, okay, C, T, Z, okay, G, T, sorry.
No problem.
So what we have talked about DNA, chromosome. Okay. So inside the DNA there are sequences like that. So if you consider one line as one of these nucleotides, purine pyrimidines, then there are sequence, okay? There are sequence, okay? Suppose, we are supposing, okay, there are lot of millions and billions of sequences is possible, but we'll see for our importance what does that look like, okay? So, set of three sequence, okay? This is a gene, okay? We have that sequence nucleotide inside that DNA. There is this number of nucleotide. We call it purine and pyrimidine. These are adenine, cytosine, thyrosyl, and guanine and uracil. Okay? So these are arranged in a sequence. So this is called gene. Okay, a set of three is called gene. Every three set is a gene. Okay, so this is a gene. So as we have already explained, this is a cell, eukaryotic cell. Then inside the cell there is chromosome. So it is inside the chromosome. These are DNA, and then these are sugar phosphate backbone. That is like a structural for integrity. And then there are, as I have said, A, G, T. This is depicted in color. Okay, yellow, green, orange, and blue. So these are the genes. If the set of three nucleotide is one gene. So before going further, I'd like to mention this is the logo of Human Genome Project. So this was conceived at 1990 especially funded by US government to sequence all of the genes in the human, and it was completed. It's not as such completed yet, but it was declared complete at 2000. But still, only 92.1% of the genes were covered, and rest 7.9% were not covered. And this was completed almost with some 79 or 80 missings around 2020. So this is the logo. They have mentioned everything. This is biology, physics, ethics, informatics, engineering, and chemistry. This is Human Genome Project. So in this picture, this is a chromosome. This is one of the chromosomes, and these are the numbering from here physical map, and there is number of base pairs. So inside the chromosome also there are various regions of the gene, where one of the region would be active, more active, another region would be less active, and there are portions which is called exons and introns based on the activity level. Okay, so I think I have someone from computer science background, binary system. Okay, when I knew this concept that everything works. You also IT. So in computer only understands 0 and 1. This is binary system. Okay. So can you imagine you are making an image picture of something, and that is stored in computer as 0 and 1. Yes, binary system. Binary system. Like if you say 1100, that is 28. Like that, binary system. So similar to the binary system, gene also works in this system, because that nucleic A, T, C, Z sequence in 333 set, I have said that is like a code. 1 byte. That is like a code. Yes, that is like a byte. That is we have also byte Dalton KD. We have that measurement system in that genetics also. And then this is like a code. Same thing happens with the gene. So this is backbone where you support these genes, and these are the nucleotides. Okay, so for example, the main function how gene works is gene works by sending signal to produce specific kind of protein, and protein functions not only protein but other things also. Especially protein is more important. So enzymes is protein, structure is protein, our skin has protein, our everything has protein. So basic structure of our body inside the cell and outside the cell protein is most important thing. Yeah, we have other things which are equally important, like we have phospholipids, we have other kind of like phosphate backbone, they have that nucleic sugars, everything is important, but this is focused basically for protein, because the production of other material from precursor is also based on the protein, which is enzyme. Okay, so one enzyme makes one kind of material into another one. Enzyme is always, and most of the time, 99% of the time, is a protein. So this is like binary system.